the recording. Last time we were talking about, and it's probably sharing the screen would do. Last time we finished with the gymnosperms and we kind of overviewed the few major terms, right? The haploid, single set of chromosomes versus diploid, double set of chromosomes. And remember, haploid set of chromosomes would be found in the sperm or egg, so gametes. Then the spores, remember the spores are always haploid, right? And then gametophytes, the one which producing sperm and egg, and then plants. And again, remember, plants are weird comparatively to the animals, because usually we're talking about meiosis producing egg and the sperm, and from the diploid organism it goes into the haploid state. That's for animals. Plants are doing their own things. They are very, very different. If you think about alien life on Earth, that's pretty close to that, because they have a very different, like, for example, very different strategies, like animals, right? In order to interact with the environment, we're moving into the environment. Most of the animals, not all, but most of the animals have active phase. Plants do not do that. They grow into the environment and exchange whatever they need to exchange with the surroundings, so to speak. All right, now diploid organism, double set of chromosomes. Remember, from mother, father, we talked about that before, from male, female. Now zygote, this is basically a fusion of egg and sperm, gives us the zygote. Zygotes start dividing. In plants, it will lead to the seed. And seed, of course, will have technically three major parts. The embryo, which is a diploid. The food source for embryo, that would be a triploid, and we'll talk about that later on. And then the seed will grow into a sporophyte, right? But the seed will have a third very important part, is the cover. And in angiosperms, the one which we'll talk about today, they will have something which known as the fruits surrounding, the vessel surrounding the seed, so to speak. And this is what angios means, the vessel surrounding something. Now, dry land adaptations. Talk about that last time. Talking about today means what? It will be on the test. Woohoo! And the test is what? A week from now, test number 2. I will tell you right away, historically, plant unit, test number 2 in bio 1, talking about the photosynthesis, cell respiration, test number 2 in bio 2, talking about plants. Historically, test number 2 is the lowest grade test, and that's one of the reasons why one test grade will be dropped by the end of the semester. Now, dropping the test, what does it mean? Very simple. You'll still see it on the blackboard. You will. Yeah, I'm not going and erasing the lowest grade or anything like that. No. But in the blackboard, instead of 4 tests, which will count toward your grade, I will say drop or do not count toward your grade 1 lowest test. So you will have only 3. Okay. So you will still see it on the blackboard, but it will not count toward your average. And once again, it will happen after you will take and I will grade test number 4. So, and I'll send the announcement, all that particular stuff, and you'll have any questions. Yeah, we discuss it later on. So dry land adaptations will be on the test. Obtaining adequate supply of water and transporting it. So this is why vascular system is extremely important. Development of true roots to take the water out from the ground and send it to the shoots. This is where the photosynthesis usually takes place. Well, shoots and the leaves, etc. Then the means of physical supports. Remember that the plants are growing into the environment, so they have to go literally against the gravitational pull. They're going against the gravitational pull of the whole planet. So they need to do that. They need to somehow reinforce those particular structures, the stems. Initially, remember, the mosses are relatively short. Why? Because they cannot grow too tall. They start breaking down, not enough physical support. Nowadays, we have the gymnosperms and angiosperms growing literally hundreds of feet tall. Why? Because they do have that means of physical support. Yeah, conducting tissues now get sperm to the egg without the water. Initially, they started with the water, but dry land. That's basically the water was the echo from the algae ancestors. Algae, of course, lives in the water, plenty of water, so the sperm could swim no problem. Now, dry land, and that's dry land adaptation. So sperm would be surrounded by the very specific, very tough shell to protect it from what? From dehydration. So now we start looking at the pollen, right? Wind-blown pollen grains. Now, protective structures for developing zygote. So zygote, in order to develop, it have to have a food source and then the protective cover from keeping it from drying out. Those tender tissues can dry it out relatively fast. Therefore, we start looking at the evolution of seed. All right? Yes, protecting and nourishing the embryo. And then, of course, we have a less carbon dioxide in the air because at some point of time, plants used up a lot of it. Now they come up with a very specific structures known as the stomatas, and we'll talk about that hopefully today. And then, well, we'll talk about the withstand of a white waxy cuticle and so on. So, and again, waxy cuticle to prevent from dehydration. So today we're talking about flowering plants. Anthophyta, Angiosperms, Magnolia phyta. Either or. Magnolia is considered to be one of the simplest from the evolutionary point of view flowering plants. Therefore, either or will work just fine. Flowering plants, Angiosperms, or Magnolia phyta. So, vascular seed, vascular plants produce flowers, hence the name, and now seeds enclosed within the vessel, angios. So, and sperm is a seed. So, seed within the vessel, seed enclosed within the fruit. Not just having a cover, it has the other thing known as the fruit. Okay, that's the main difference between the gymnosperms and angiosperms. By the way, most likely would be a question. Briefly describe the difference from the seed point between the gymnosperms and angiosperms. Ta-da! And we already talked about that it's the most diverse, successful group of plants. Flowers, since they are flowering plants, may contain usually something which is known as a full flower or complete flower, will have 4 major parts: sepals, petals, stamens, and carpels. Sometimes carpels referred as the pistils, right? That's why you shouldn't be messing with the flowering plants. They have pistils. Yeah. Biological, according to biological. My kids are called dad's jokes nowadays. So anyway, so but in many cases, a flower will be missing something. It could miss the sepals. Some of the flowers don't have petals. Some of the flowers don't have stamens. Some of the flowers don't have carpels. So we'll discuss that momentarily. So if they're missing at least one part out of those 4, that would be incomplete flower. So tracheophytes, already talked about that. Tracheos means tubes. So tracheophytes, the plants with the tubes, transporting system, vascular system, vascular plants, and we discussed the ferns, gymnosperms, and today we continue with the angiosperms. So now, ovules. Ovules are structures within the ovary, and within the ovule, usually we have the egg. So we have ovule. Within the ovule, there is an egg. Sperm comes in and fertilizes it. So ovules will become a seed. Ovules will be enclosed or located in the ovary. Okay? ovary, overgrown ovary becomes a fruit. Think about it, that sometimes we see like a peach, right? Or a pear, well, not pear, plum or apricot. We have one seed per fruit, a large or avocado. One seed surrounded by the fruit. It means that it has only one ovule with one egg, and therefore one seed in the fruit. But if you open a pear, cut open the pear, or strawberries or apples, or any kind of citrus, orange, tangerines, or anything like that, there are usually a multiple of seeds there. Meaning what? In one fruit, in one ovary, there was several eggs, several ovules. Those ovules has been fertilized, so now we have several seeds within a single fruit. And we'll talk about next time. I'll confuse you about the types of fruit. We have a simple fruits, and we have compound fruits, and we have aggregate fruits. We'll talk about that later on. Don't worry about that. So yeah, at least right now. So life cycle, same kind of thing. Same thing means what? We have a sporophyte and we have a gametophyte. Now gametophyte are drastically reduced, literally a handful of cells. The male gametophyte essentially is 2/3 cells, depending on the stage of the gametophyte, but 3 cells, 2 sperm and 1 something which known as a pollen tube cell. And then the female gametophyte, 8 cells. So from the dominant part of life of the plants in the non-vascular seedless, mosses, and liverworts, the gametophyte drastically reduced their size, and they are completely depends now on the sporophyte. So if you're looking outside, I'm looking outside right now and I see the oak tree. If I see it with my naked eye and I know it's a flowering plant, here we go. That's the sporophyte. The sporophyte generation is dominant. Now, the plants continue to be heterosporous. What does it mean? Remember, we talk about different spores. Spores are different size. Okay? Male spores are much smaller than the female spores, and the spores, of course, are haploid. Now, microspores, male, and they essentially will be turning into something which we know as a pollen grain, and megaspores will be located deep inside of the flower. Today we'll talk about that, and next time on Tuesday we'll discuss some of the aspects of the reproduction of the angiosperms. Now, in this particular case, on a reproduction of flower plants is something which known as a double fertilization. 2 things will be fertilized almost at the same time. It will be on a test. Briefly describe the process of double fertilization in flowering plants. I will introduce it right now, and I will talk about that much in a greater detail on Tuesday, next week, essentially. So double fertilization, what does it mean? Within a female gametophyte, there is 2 major cells. There is 8 cells total, but there is 2 main ones. One of them is the egg, another is the central cell. So far, so good. And there is antipodal cell, and then there is 2 more cells, which is right next to the egg, but they're important, but we're not going too deep into that part. So we're looking at the 2 very important cells for the double fertilization, the egg and the central cell. So now what exactly is happening? Pollen getting on top of the flower, start growing, and it's produced. Each pollen grain contains 2 sperms. So sperms are slowly traveling through the flower, and eventually the first one will meet with the egg. Ta-da! They merge together, they fertilize, well, the sperm fertilize the egg, everything is good. So now we have a first step to the sporophyte, we have a zygote. But we have a second sperm coming in. The egg is already fertilized, and the sperm like, well, I need to fertilize something, and it goes to the next best thing, which is known as a central cell. And it fertilizes the second cell, the central cell. That's why it's called double fertilization in the flowering plants. Now, the outcomes of that double fertilization, the zygote will start dividing, growing into the embryo. And then the central cell now divides, and it will be produce a food for the embryo, the actual food source for the embryo, the seed, for the seed. So here we have the outcomes: diploid, zygote, double N, and triploid endosperm. Just kind of introduction, throwing it there. We'll discuss that next week in a greater detail. So here we have a life of angiosperms, and in this particular case goes pretty much the same thing. We have a sporophyte generation, diploid one. So in this particular case, deep inside of the flower, the egg will be growing, and again, it will go through the several divisions. We'll talk about that soon enough, but the end result, as you can see, 1, 2, 3, 4, 5, 6, 7, 8 cells. As a matter of fact, it will be 8 nuclei and only 7 cells. The central cell will have 2 nuclei, so it will be dikaryotic. So anyway, within the enters, we have the microspores produced. Microspore is produced. Later on, it goes through the process of mitosis, creating 2 cells. Immature will contain the pollen tube cell and the sperm cell, and then the sperm cell will divide into 2 sperms. So during the pollination, whenever the pollen grain will land it, 2 sperms will start traveling down that particular pollen tube. And again, 2 sperms coming in. One of them will fertilize the egg, and the second one will fertilize the central cell. That's the very important part right here with the outcome endosperm. Triploid. Why triploid? Because the central cell had 2 nuclei, and we're adding 3rd nuclei from the sperm. So 3 nuclei, haploid nuclei merging together, creating triple set of chromosomes. And then, of course, we have the zygote. All right, moving on. So we talk about that, green algae, modern charophytes. Then they start, charophyte stays in the aquatic environment in the water essentially, but some of them move to the dry land and come up with the non-vascular plants. Then we're looking at the non-vascular plants at some point of time, modern non-vascular plants. That was the ancient non-vascular plants, come up with the means of transporting the chemicals. Which chemicals? Water to the leaves and sugar from the leaves. So again, photosynthesis will be on that particular test as well as a lab practical. So they come up with the seedless initially. So looking at the seedless vascular plants, and then we start having, well, they start having a problem of not enough water present, so the sperm cannot swim to the egg. As a result, they had to come up with the wind-blown pollen and the seed to protect the embryo from dehydration. And here we're looking at the gymnosperms, ancient gymnosperms, and the ancestors of the gymnosperms, modern gymnosperms, and angiosperms. And angiosperms come up with the flower and the seed, of course, and the flowers and the fruits. So distinguished characteristics will be the flowers, not always flashing, not always bright colored or anything like that. We'll talk a little bit about that. And then finally, overgrown ovaries, that's the fruits. So if you'll think about it again, remember I told you that we are horrible to plants. So flowers are the organs of sexual reproduction of plants, of specific group of plants. So whenever we are cutting that, what are we cutting? We are cutting the organs of sexual reproduction for the plants. And then whenever we're eating, consuming fruits, what are we eating? We're eating essentially overgrown ovaries in most of the cases, if we're going for flesh. If we're going for the seeds, we're eating the embryos and the food source for the embryo. So once again, covered seed. Why it's called covered seed? Because the seed within the vessel talking about fruit. It has, all of the seeds has a cover to protect the embryo. Gymnosperms or angiosperms, but the gymnosperms don't have the fruit. Make sure you understand that. Now, some of the examples of what we are using the plants for, well, besides food, right? Remember I told you, food or medicine. So here we go. Some of the things which we are using the plants for, most of the time, well, all of them are flowering plants. Belladonna plant pupil has the atropine, the dilation of the eyes, foxglove, menthol, of course, ingredients in the cough medicine to relieve that pain reliever, opium poppy, right? Morphine, heroin, essentially. Yes, great pain reliever, but also extremely addictive. And you know, we have a huge problem here in the United States, pretty much worldwide, with the opium or opiates abuse. You're kind of getting used to that and you want more. Then the malaria preventive, very important. Remember that the malaria are #1 disease killing humans from the, well, with the protists. And then ovarian cancer drugs or cancer drugs, we were trying to see, or some of the plants have them, curare muscle relaxants during the surgery, and of course at the high concentration of the turbocurarin, all of the muscles can relax and the person can die, and so on and so forth. But yeah, we have quite a few usage for fruits, well, for food as well as the medicine. Now, the classification of the flowering plants, again, very large group. And in this particular case, as you can see, it has quite a few major groups, but two of them, the largest one, known as the monocots and dicots. Mono and dicot. Mono means one, di means two. Now all we need to do is figure out what the cot is. In Russian, cot means cat. Yes, cat, male cat specifically. Male cat will be cot, but that's not Russian. So it's it comes, the cot is abbreviation from Ketillidon. So we have the plants with one Ketillidon, and we have plants, flowering plants with two Ketillidons, right? We can say, okay, well, makes sense, but what the heck is the Ketillidons? Ketillidons is embryonic leaves, nothing else. Ketillidon equal embryonic leaf. So if the embryo has only one leaf, that's the monocot. If the embryo has two embryonic leaves, whenever you're looking under the microscope, that's a dicot. See how easy it is? Yeah. All right. So that's basically kind of classification. So we will pay our attention to mostly monocots and dicots. Everything else, yes, they are important, but we are not in botany. We are briefly touching, so I don't have time to go about the water lilies and the star anise and then magnolias. They're really interesting, very interesting group, but I don't really have time to go over all of them. Okay. So again, if you'll be able to recognize monocots versus dicots, and again, monocots and dicots will be on the test. Make sure you know the difference between the monocots and dicots. And what are the difference? So here's the main difference, and you will see it in the lab next time. But here we're looking at the seed, and within the seed, there is only one embryonic leaf, monocot. Within the seed, we have 1 and 2 embryonic leaves, dicot. Whenever you're looking at the leaves, if the veins in the leaves are running more or less parallel, that's most likely a monocot. If it has something else but the parallel venation, something which is known as a palmate, or what's the next one? reticulate one, that would be the most likely a dicot. Now, vascular bundles. Remember, we're talking about the tissues. If they are, there is two types, phloem, xylem. It's moving different ways. So if those particular vascular bundles scattered throughout the stem, that's the monocot. If they are arranged in a nice ring, and today you will see it in the lecture as well. I'll show you the slides and on next week. Yeah, next week, lab number 9, we'll talk about that. You'll see that as well. So if the vascular bundles, the bundles of the vascular tissue arranged in a nice ring, that's most likely a dicot. Whenever you're looking at the roots, if the roots are kind of all throughout, known as the fibrous roots, you cannot tell which one is the main or so-called tap root, then it's most likely a monocot. The dicots, most of the dicots will have a main root with the secondary roots coming out. The pollen grain will have only one opening in the actual pollen grain. The pollen grain right here will have 3 typical openings. And then whenever you're looking at the flowers, in the monocots, the flowers typically have a multiples of 3. For example, 6 petals, the parts, or 6 stamens. The part would be or 3, 6, 9, and so on. So multiples of 3 for the flower organs and multiples of 4 or 5 for the dicots. So let's say here we have a dog rose, right? That's the rose. Wild one. 1, 2, 3, 4, 5 petals. If it's a 5 petals, most likely we're looking at the dicot. And then we're looking at the leaves, and we see that it has non-parallel venation, so it's definitely a dicot. Make sense? All right, so we'll finish that particular PowerPoint. Let me pull out the next one, which is this one. Yes, so now we'll talk about the Formant Function, and so basically organs of the plant. Again, a really nice, really quickly, the cladogram showing you the vascular seeded plants. So we have evolution of seeds, and then Dicots, we discussed that, and then ginkgos with a single species nowadays, coniferous pine trees and such, and then Gametophytes, specifically Welwitschia, Gnetos, Gnetum, and Ephedra, and then finally the Angiosperms. So again, talk about the monocot and the dicots, and this is basically the Angiosperms. We can see monocots and the dicots. So major groups are based on number of Ketillidons on the embryo, and essentially it's a embryonic leaves. One embryonic leaf, it's a monocot. 2, that would be a dicot. All right. So Ketillidons, the seed leaves or embryonic leaves. So here we have the picture, and this is the embryo right here, and that's a single leaf, 1 Ketillidon. On the dicots, you can clearly see that's the embryo, and now we're looking at 1 and 2 Ketillidons. Those leaves are really important for the embryo because they're helping in digestion of the stored food for the embryo. For example, here it's a kernel of corn. In corn, as you know, corn is high or rich in starch. Starch is a large molecules basically made out of thousands upon thousands molecules of glucose. So before the embryo can digest it, take it and start building whatever it needs to build, it have to, the large molecule of starch have to be broken down into a glucose molecules, and this is what the Ketillidon does. It breaks down and feeding the embryo with the simpler molecules. Same thing, and that's definitely some sort of bean. Beans have a high level of proteins, and you know by now proteins are the chains, hundreds upon thousands of amino acids combined together. Again, in order to get the amino acids, the Ketillidons breaking that particular proteins and feeding the embryo. So 1 or 2 Ketillidons. Now, embryo with a single Ketillidon, embryo with double Ketillidons. Once again, there will be a question on the test, compare, contrast monocots versus dicots. Make sure that you know several differences or major differences. Pollen with a single pore, well, with a single pore, 3 usually pores for the dicots. Flower multiples of 3, flower multiples of 4 or 5. Question is, let's say you're counting the flower parts and you ended up with 12. You counted petals, there is 12 petals. Is it a monocot or dicot? Well, it's a half cot. 1.5. Mono half cot. What do you think? Any ideas? Multiples of, well, once again, multiples of 3 for the monocot, multiples of 4 for dicot. Or you are finding the flower which has a multiple, well, you're counting and it's 15. So it could be a multiples of 3 and multiples of 5 at the same time. So what is it? How can you determine if it's a monocot or dicot? Nobody knows. Simple, very simple. What you will need to do is look at something else. If the flower is confusing, then you're looking at something else. Like what? If you have a seed, you can look it up how many Ketillidons it has. If you have leaves, you can see veins are parallel. Most likely, then it would be a monocot. Major leaf veins are reticulated. We'll talk about that separately. Then most likely it's a dicot. Then you can cross the stem and look at the bundles. If the vascular bundles scattered throughout the stem, that would be a monocot. They are located in a ring, that would be a dicot. Roots, you can pull it out and the roots are coming out adventurously, so to speak. That would be the monocot. If the roots have a major root and then the secondary roots and tertiary roots coming out, then it's most likely a dicot. Secondary growth absent, meaning that they are not really trees. You're looking at the tree with a very large trunk, that's most likely a dicot, and we'll discuss that all together today. Very briefly though. All right, so here, yes, the same picture, already talk about that. So now, tissues. Let's kind of talk about the tissues. As any kind of tissues, animal or plant, there is a simple tissue which will have only one type of the cells, or complex tissues which have several different types of the cells. As the regular plant cells, they will have a primary cell wall. Remember, cell wall made out of cellulose, and the primary cell wall will be penetrable by any kind of chemicals. It has some openings, so basically it keeps the cell intact, and then within the cell you'll have a cell membrane. In addition to cell wall, all of the cells will have a cell membrane, and then all kinds of organelles, chloroplasts most likely, and so on. Nucleus, and then the plasmic reticulum, Golgi apparatus, etc. In many cases, the plants will produce something which is known as the secondary wall. Secondary wall, so we have a plasma membrane or cell membrane and the primary wall. All of the plant cells have that. But occasionally there will be deposits of something in between the cell membrane and the primary cell wall, and that one known as a secondary cell wall. And in some cases, secondary cell wall will the cell can be still alive inside of it, so it will be penetrable by any kind of chemicals. And in some cases, it will isolate itself and die inside, just leaving basically the skeleton, and we'll discuss that momentarily. So plant anatomy, very simple. In humans, we have all kinds of organs, we have different parts, etc. In this case, they have only 3 organs, essentially. They have a stem, leaves, and the roots. That's it. There is some parts, and we'll discuss some of those ones. We'll talk about the terminal buds, we'll talk about the leaves coming out from the stems, from the nodes, lateral buds, and the roots, and so on. But generally, 3 major organs: stem, leaves, and the roots. Okay? So now, a little bit more about the tissues. Meristem. Think about it as the embryonic stem cells. Right now, periodically, if you're listening to any kind of advertising, I heard it a couple of times. I don't watch much of the TV, but in this particular case, I heard like, well, we have such and such night cream infused with embryonic stem cell tissue or plant tissues. Well, basically what they do is taking the meristems, embryonic tissue of the plant capable of rapid growth. Usually the meristems will be located at the tips, known as the apical meristems. And the apical meristem, whenever it grows, it could be at the tips of the shoot and the tips of the root. So it will start growing. So the shoot will grow in and grow out, and the roots will grow in. So they are responsible for increasing in length. All right. That's known as a primary growth. Apical meristems grow in length, also known as the primary growth. That most likely will be a question on the test as well. Compare, contrast apical versus lateral meristems. So at the tips, and they start growing, increasing the length. Lateral meristems, sideways. They are located on the sides of the stem, for example. As a result, whenever they're growing, they're increasing the diameter, the girth of the plant, of the stem, of the trunk, so to speak. So apical meristems increase in length. This is how any kind of tree will grow tall or go deep into the ground due to apical meristems. Whenever the tree is getting thicker, increasing its diameter, that's the lateral sideways meristems, which are increasing, which also known as a secondary growth. And usually those particular lateral meristems will produce something which is known as a wood. And we'll talk a little bit about that. All right, now tissues. We have in animals, we'll have a special part of the lecture when we will be talking about the tissues and different types of the tissues. And we have the epidermal tissues, right? And we have connective tissues, and we have a muscular tissue, we have nervous tissue. That's animals. Plants slightly different. They have a dermal tissues. Same thing as us, the outer protective covering. Usually a single cell layer, usually, not always, but typically the epidermis, a single layer of cells. Now, vascular tissues, conductive tissue, right? So we have dermal tissue and vascular tissue. Vascular tissue usually goes kind of in the middle of something, in the middle of the stem or anything like that. So outer part, and then the one which is responsible for transport of materials within the plant. So if it's not dermal tissue, if it's not vascular tissue, everything else in between the dermal tissue and the vascular tissue, known as a ground tissue. This is what produces most of the photosynthesis, additional support, storage, or anything like that. All right, ground tissue can be subdivided into 2 different areas where the ground tissues are located at, that known as a pith inside of the vascular tissue, usually within the bundle. I'll show it to you today. Don't worry too much about that. And the outer, if you have the vascular bundle in the middle of the stem, so whatever is within the vascular bundle is a pith, whatever is outside known as the cortex. I'll show you the picture, don't worry about that. So in this particular case, let's take a look. Here we have a dermal tissue, epidermis. Here we're looking at the vascular bundle, because two types of the vascular tissues, xylem and phloem, I told you about that last time, they kind of pairs up. Xylem typically has a larger diameter cell. Why? Because it will have to take water from the ground and send it all the way up. High pressure, really high pressure. As a result, the diameter is usually larger. And then there is a phloem right here, smaller smaller cells, and they goes typically the movement of the water with dissolved sugar downwards, so along with the gravitational pull, so usually smaller cell. Now, from the ground tissue, we can see three different types, very different. So we have, again, vascular tissues, dermal tissues, epidermis. Vascular tissue will be subdivided into two categories, xylem and phloem. Ground tissues will be subdivided into three different tissues: parenchyma, collenchyma, and sclerenchyma. Okay, H is silent, so it's a chyma. All right, so let's move on into a ground tissue. We already talked about the parenchyma, collenchyma, sclerenchyma. Each one of them are very different in structure. Some of them are alive, some of them already, as the mature tissues, dead cells. You can say, how in the world the dead cell can make a tissue? Well, plants, remember, pretty much all of our tissues are alive, except the epidermal or top of the epidermal tissues, which are dead cells. We covered by the dead cells, right? Everybody understand that. The skin, the top layer of the skin is dead already. That's dead cells. They covering, they helping for what? For dehydration, helping us not to lose much water. But they already dead. That's why we're shading them constantly. We'll talk about that later on. So anyway, so in this particular case, some of them are, again, alive, some of them are alive, some of them dead. And then the main part or main difference is a very distinct cell wall structure. Parenchyma has only primary wall, the collenchyma and sclerenchyma has the secondary wall. And we'll discuss that momentarily. I'll show you some of the pictures. So parenchyma, that's the soft primary growth of roots, stems, flowers, known as the parenchyma. Very small cells, typically very fast dividing, have a thin primary wall, permeable to the water as well as any kind of chemicals, specialized for the photosynthesis. Again, many of them are fast dividing, so meristem, so to speak, can be used as a carbohydrate storage, short distance transport, not a long distance, because for the long distance we have a special structures or special tissues known as a vascular tissue. Parenchyma cells typically alive, okay, usually alive and capable of dividing whenever they mature. And this is the picture of the parenchyma cells. That's a typical plant cell, which you've probably seen under the microscope. We see the nucleus, we have the vacuole, you'll see the cell membrane, and then of course a relatively thin cell wall. All of them will have cell wall, but in this particular case, it's a relatively thin. The water and the solutes can be exchanged between the cells. Okay, all right, any questions, any problems? Hopefully not. All right, moving on to the next one, and then we are talking about collenchyma. Collenchyma main function is a flexible support, and the flexible support in this particular case, it develops a thick primary wall of cellulose, and then there is something which known as a pectin, which can stretch and then return as the cell elongates. Flexible support and actually to the certain degree movement. You can say, how in the world it's moving? Okay, well, think about it. Let's say everybody can visualize sunflower, right? Why it's called the sunflower? Well, first of all, it looks kind of like a sun, but if, let's say, you go into a sunflower field in the morning, early in the morning, the sun comes out, all of the sunflowers will be facing east, where the sun is coming out. Everything is nice. Taking a big, well, looking at the sun. So now, what exactly happens? Let's say you will go back to the same field, but at the end of the day, when the sun is mostly to the west, you will see that the sunflowers now facing to the west. They still facing the sun. They don't have muscles, but they are moving during the day, maybe not fast, but they're moving with the movement of the sun, so they can turn. How that thing is happening? Basically, what we'll see that there will be a lot of, whenever the part which is exposed to the sun would have a lot of sunlight, and therefore lots of water will be pumped in. Okay. Now, whenever the sun is moving, now this part has a lot of water moves in, and therefore less water right there, so it will turn a little bit. And the Collenchyma plays a very important role in that. So again, cells develop a strong, thick primary wall of cellulose, and the pectin helps with the stretching. So whenever, and the cells stay alive and metabolically active, typically, even though they have a very thick cell wall. So now here we're looking at the same, well, at the Collenchyma cells. As you can see, the cell wall is much thicker comparatively to the parenchyma. Same part have vacuoles, have the nucleus, but now in many cases the cell wall, or most of them, will be much thicker, flexible support. And finally, sclerenchyma provides the mechanical support and the protection for mature plants. And the sclerenchyma cells initially alive, but later on they will develop thick secondary cell wall. Remember membrane, primary cell wall. Now they start putting additional stuff such as the cellulose and lignin, where they will lignify those particular secondary cell walls. As a result, essentially the cell is isolating itself. It's building up. It's creating the cage where it will die inside, because that particular secondary wall will prevent it from exchanging of the nutrients and the waste. So sclerenchyma as the adult cell is the dead cell. Essentially, it's a skeleton of the cell. It builds up a very thick wall, and it's used for the mechanical support now. That's it. Two types, sclerids and the fibers. You probably can figure that out. The fibers, sclerids. Anybody tried the peach before? Pear, sorry. Peach too, but pear. Whenever you're whenever you're chewing on a pear, you will feel tiny little bitty kind of like grains. That's the sclerids you are essentially eating. Now, sclerids also building up the seed coat for many of the plants, or for many of the seeds, and this is where the peach coming from. If you try the peach, there will be in the middle, there will be a single seed, very large, very thick seed coat made predominantly out of sclerids. And then, of course, fibers, any kind of wood fibers, anything like that, that's elongated cells which are resist to stretching, unlike the Collenchyma. So here what we're looking at, this cell is still active. It has a nucleus right there, it still has a vacuole, but look at this red stuff. This is the cell walls. Cell walls provide a very rigid support as well as the protection, and you can see that that particular cell completely isolates itself from the other ones and dying off. So the cells are eventually, or mature sclerenchyma is mostly a dead cells. As a matter of fact, most of the plant is a dead cell. So whenever you're looking at the oak tree, for example, 95% of the oak tree are already dead cells. It's not walking dead, it's kind of standing dead. Not necessarily, but yeah. In a large oak tree, about like 2 inches on either side in diameter, that's the live cells, and of course the live cells of the leaves. But the stem itself, pretty much dead cells. So here we're looking at the something which is known as the sclerites, tiny little bitty granules, and you can see that that cells is already dead, a very thick cell wall. That one is probably alive but about to die because it built up a very thick secondary cell wall. It has the cell membrane, cell wall primer, and then this one is building up a secondary cell wall. And here we're looking at the fibers. So plant tissues continues, and we talk about now we are talking about vascular tissue. So we're done with the ground tissues. Now we're moving with the xylem. Xylem is the tissue which conducts water and minerals typically upwards from the roots. The water moves upward to the leaves. Why? Photosynthesis. Remember, everything based around 2 of those particular reactions will be on the test. So in this particular case, water will have to move into the photosynthesis. Photosynthesis occurs, everything is fine. Now sugar is produced. Now the sugar will be traveling downwards, right? So to distribute throughout the body of the plant, because those cells are in need of the sugars as well, in need of food. So the red arrows right here showing you the movement of sugars, organic compounds, food, and that would be the phloem. So xylem conducts water and minerals from the roots upward. They are reinforced, the cells or cell walls reinforced with the lignin. As a result, the cells inside are dying. So basically it becomes a tube and the cell inside dead. So the water is moving out. So cells are dead in the xylem. All right. Now, whenever we are looking at the xylem, you will see that the very large openings. This is the vessels, and those particular vessels, this is where the water coming. Again, very high pressure of the water, because again, we are talking about that the water have to be pumped from the ground all the way to 10, 15, 20, 30, 40, 100 feet high. A very large, well, a very high amount of pressure in those particular tubes. So they're from the collapsing, they have to be reinforced, which makes those particular cells die. So vessels are typically, whenever you'll see a large diameters, this is when most likely you're looking at the xylem. So now phloem. Phloem conducts sugar from the leaves, and photosynthetic parts usually are downward. This is where you will have the photosynthesis, and then it moves out. Cells are living, full of sugar, so they're happy, and they slowly conducting that particular chemical. All right, very, very simple. So in this particular case, again, kind of tubes, but between the tubes they have a sieve plates, and this is how the high concentration sugar will start moving right here, and so on and so forth. So everything is alive here. Now, whenever you're looking at the cross-section of the stem, and that's kind of a representation of the cross-section of the stem. So what we're looking at is the xylem and phloem combined together. They kind of running parallel. The water goes up, the water goes down. So that creates the vascular system, and that creates something which is known as a vascular bundles. Because now you have a very large diameter cells known as the xylem, and bunch of small diameter cells. It's not a really good picture. It's a small diameter, but a lot of them, and that would be a phloem, together creating vascular bundle. Remember, we're talking about the ground tissues, vascular tissues, and the dermal tissue. So here we're looking at the epidermis. Here we're looking at the vascular bundles, everything in between known as a ground tissue. All right. So again, we're looking now at cross section. We're looking at the xylem, moving the water and the minerals upward. The water can exchange at any point between the xylem and the phloem, of course, transport of the sugar from the photosynthetic parts all the way throughout the body. Now, whenever you're looking at the micro photograph of that, you can see this particular combination. That's a vascular bundle, a single vascular bundle. Look like a monkey face, right? Or alien face, right there. See the eyes, I guess the nose, and then kind of hair, whatever. So in this particular case, the large diameter vessels, right here, right there, that's a xylem. Here we're looking at the smaller diameter vessels. That's most likely a phloem. Okay. So now again, remember vascular tissue versus the dermal tissue versus the ground tissue. So dermal tissue, vascular bundles, and then everything in between known as the ground tissue, specifically pith. In this particular case, we're looking at the picture of monocot. How can I tell that it's a monocot? How can you tell that it's a monocot? Look, those particular vascular bundles are scattered throughout the stem. They're not arranged in a nice ring. They scatter throughout the stem. And because they scattered, that's I can tell that it's most likely was a monocot. Now look, the xylem is facing inwards, typically. The phloem is facing outward. In between here, there will be something which known as a meristem, which producing xylem and phloem. So here we have a phloem, the xylem, and the part in between of them would be the meristem. So here, whenever we're looking at the closer look, so vascular bundle, large vesicles would be a xylem. And then we have a smaller one would be a phloem, living cells, non-living cells. And in between of them, there will be a layer of fast dividing cells. Here, whenever they are specializing inside of the stem, they'll specialize into a xylem. Outside, they will specialize into a phloem. All right, so here we're looking at the pictures of the dicot versus the monocot stem. See, those particular vascular, the purple vascular bundles scattered throughout the stem. They scattered throughout the stem, that's a monocot. They arranged in a nice bundle, a ring around that, that would be the dicot. Now, on the ground tissue, here we have the dermal tissue, vascular bundles, right? Neatly arranged in the ring. So whatever is between now the ring of vascular bundles and the dermal part, so that layer, that known as the cortex, whatever is inside of that ring would be known as the pith. Now again, we're looking at the phloem, we're looking at the xylem, and quite often you will see the additional sclerenchyma fibers to support, to provide the mechanical support for the vascular bundles. All right. So now, if we're looking at the dicot stem first year, and then we're looking at the dicot stem second year. So now we start seeing that it's increasing in diameter because the cells in between the phloem and the xylem continue to divide. And whenever they continue to divide, they will create the secondary phloem. They'll create the secondary xylem. As a result, it becomes thicker. So whatever is inside is already dead. It's no longer functioning, so to speak. It still functions as a vascular tissue in most of the cases, transporting, but there is no living cells or almost no living cells in the middle of the stem, specifically in the middle of the, let's say, tree trunk. All right, moving on. So here what we have, you can see a nice ring, a middle part known as a pith. That would be a primary xylem. That's a secondary xylem. So that particular stem is 2-year-old. The pith at this particular moment is still alive, but later on it will die off. And here we're looking at the phloem, and that particular thin layer right here between the phloem and the xylem is actually vascular cambium or vascular meristem, the cells which will produce phloem and the xylem. How old is this stem? We already talked about it. This is a 1 annual ring. This is the second, so it's on the second, well, basically on the 3rd year. So cambium, just talk about that. Meristem, it's a vascular meristem. Those cells are dividing. If they are inside, they will give or they will create the xylem. If they will outside of that particular vascular bundle, they will specialize into a phloem. So yeah, vascular cambium, we can say vascular meristem, whichever it is, same thing. And that's a lateral meristem, of course, because it's on the side. It's on the diameter on the side. So that's a lateral meristem. Here what we'll have, and we already talked about the cortex. Cortex is between the any ground tissues between the epidermis and the vascular bundles on the outer side. So here whenever we're looking at the cross section of the stem, we're looking, let's take a look. The bark, right? Bark is already dead cells. They are lignified, heavily lignified to provide the protection from any kind of environmental things, right? Then there is a bark cambium, the meristem which produces bark. Then we have phloem, vascular cambium, so that particular layer is still alive. And then we have a xylem which is dead. All of that is dead cells, then the green and the orange is living cells, and then the gray cells, only the bottom, well, the top kind of is the living cells, everything else is dead. Now, whenever you're looking at the xylem, most of the time in a several-year-old stem, you will see 2 very distinct parts, the lighter part and the darker part of the xylem. The lighter part known as, I'm moving on a little bit further. We'll talk about the hardwood and the sapwood. The darker part known as a hardwood and the lighter part known as a sapwood. And we'll talk about that momentarily, what exactly it is. So now in a monocot stem, look, phloem, xylem, and then that probably would be the vascular cambium, the cells which are dividing, producing or specializing into a xylem and a phloem. Quite often, they will have this set of fibrous structures to reinforce the stem, and that would be, of course, the sclerenchyma. So let's take a look on another slide. So in this case, we're looking at the cork on the outside, cork cambium, that's alive. Then we have a little bit of the cortex, and then we have a phloem ray, which is still alive. We have the vascular cambium between the xylem and the phloem, that's alive. So that part is alive. The outside is dead. The middle part is dead as well. So here what we essentially see, that's a cross section of the helianthus, which is a sunflower. So you'll see the pith. You will see really nicely the ring of the vascular bundles, and of course inside of the vascular bundles, what you will see the phloem, live cells. You will see a large diameter cells, which will be a xylem. Vascular cambium will be in between of them, and then you will find the sclerenchyma of fiber cells providing the overall support to the stem. In the monocot stem, what you will see is tons of those vascular bundles throughout the stem. They're not arranged in a nice neat ring as here. And the ground tissue, also known as a pith, right here. Here you have a pith, which is inside, and the outside is known cortex. Essentially, you don't have a cortex in the monocot. All right, continue with the stems. Secondary xylem known as the wood, and the darker one known as the hardwood. And hardwood is a xylem which has been clogged up, so to speak, no longer transport water. It's darker, it has all kinds of things which literally clogged up those relatively thin vessels. Therefore, it's really non-functional as the vascular system. However, it provides the mechanical support to the tree. The sapwood, the lighter part, is still conducting. It's a functional part of the xylem, hence the name sap, right? Some sort of sap will be coming out from the sapwood because it does conduct some of the minerals as well as the water. So here again, we're looking at the cross section, and growth 3 represents the annual production of the xylem. So first year, second year production, that's a pith. And here we're looking at the phloem. Now think about it. What would happen if, for example, we will continue to have living cells right here? The living cells will start dividing. As a result, they'll start increasing the pressure inside of the stem. And if they are increasing the pressure inside of the stem, what would happen to the stem eventually? It will crack. Therefore, inside of the stem, the plants have pretty much dead cells. The outer part of that are the living cells. As a result, it grows outside, so to speak. If it will start growing inside, eventually it will create the pressure to crack the stem out. So here what we're looking at is the cross section of the trunk of some sort, and you will see the darker area known as the hardwood. The lighter area is the sapwood. Both of them are xylem, but again, the sapwood is functional. No, both of them are functional. Both of them provide mechanical support, but that one is also conductive, and that one is no longer conductive. So in this particular case, this is what we are looking at: the summer wood versus spring wood. The lines, the darker lines, why exactly is happening? Spring wood, whenever the spring comes, usually this is where the water exchange happening, the really fast division of the cell occurs. Typically it's after the winter, which was a very slow season, and we're talking about the cold winter, not like the winter we have here in the Houston area. But when it's pretty much nothing is happening during the winter time because it's too cold, and then the spring comes, the warmer time, the exchange of the materials, the cells start dividing really fast. So we'll end up with a very wide line. And then during the summer, too hot, everything slows down, the plant start preparing itself for the next winter. So as it slows down, the divisions are slowing down, and therefore you will have that particular dark line. So that's the year, so to speak: spring wood, summer wood, and then another spring wood, and then another summer wood. Remember, we were seeing last lecture, you saw the cross section of the sequoia tree with the thousands, almost 2,000 of the rings. Just imagine to count those ones. So now the meristems. This one is something which is known as a terminal bud. Terminal means at the end, right? The terminus means the end in Latin. The Terminator. All of you probably seen at least one of those particular movies. What exactly the Terminator was? It was the ender of whoever. It was sent by the machines from the future to the past to end a particular person, so he will not get the revolution going, and so on and so forth. Same thing with what's the walking, I forgot the name of the show. Walking Dead, right? Yes, Walking Dead. There was a whole season when they going toward the terminus. That was the some sort of train station or what have you. I didn't watch it. My wife was watching it. I was like, and she's like, well, they're going to the terminus. I'm like, that's a bad name. It basically means that it's the end. Literally, it's the end. The terminus means the end. And yes, apparently it was like a bunch of people who would capture you and say, well, you're either eating the other people or we'll eat you. So yeah, the end. So anyway, apical meristem. Remember, this is the fast dividing cell. So those particular cells will be growing up, not down, up. They will start increasing. So those ones are dividing. Those start growing, increasing in size. So they're pushing it up. And those particular apical meristem on the top will produce a special hormones to keep the auxiliary or lateral buds dormant. It's like, I am the terminus, I am the end of the stem, so you are hushing and I'm getting all of the nutrients because I'm dividing first. So everything goes good until something happens. For example, there is some sort of wind or anything like that, which will destroy that particular terminal bud. If it's destroyed the terminal bud, hormones are no longer, chemical messages, no longer produced. As a result, those particular buds will start growing. In many cases, and we are using it quite oftenly in the horticulture, or agriculture, whichever you prefer. Like, for example, if you have a tree and you need to prune the tree. By pruning the tree, you essentially removing the apical meristem. As a result, over a period of time, you will have a much more thicker tree, so to speak, lots of branches, because instead of one, you will end up with two, and then you pruning, you will end up with four, and so on and so forth. So anyway, that's the terminal bud. We'll talk about the terminal bud on the next week lab. So here we have the terminal bud, the auxiliary buds, and then you'll start seeing the layers within the xylem and phloem. So if you see any kind of woody plant, it has a secondary xylem, and most likely you're looking at the dicot. Herbaceous plants does not have a secondary xylem, only primary xylem, could be dicots, could be monocots. So in the root structure, we're looking at the very similar thing. We're looking at the apical meristem, but now at the tip of the root. This is the zone of division. This is where the cells are dividing. Then there will be a zone of growth or elongation. This is what pushes the root deeper in the ground. And then the zone of maturation. This is where we'll start seeing the root hairs coming out. Root hairs are very important for the plants because they drastically increase the surface area. Here, the surface area is are just that, right? Here now the surface area is all that. So root hairs are drastically increasing the surface area. Why they need the surface area? Higher the surface area, more absorption occurs. And they need to absorb minerals as well as the water to send it up into the leaf so they can do the photosynthesis. In addition to the root hairs, remember we're talking about mycorrhizae. We have the fungal threads, hyphae surrounding the root hairs, increasing it even more, the surface area that is. And then, of course, on the very tip of the root, we have something which is known as the root cap. That's already either dead or dying cells, but they are preventing or they are protecting the apical meristem from any kind of environmental factors such as sharp rocks, the sand grains, or anything like that. So in a cross section of the root, what you will see is the xylem and the phloem, right? That's a vascular bundle. Now you see the epidermis. Now we can clearly see a really nice cortex. That's the dicot root. In the monocot root, what you will see is the endodermis, essentially a ring of the endodermis, but you will see the xylem and the phloem. You will see the epidermis, you will see the cortex, and now quite often. In the monocot roots, you'll see the pith as well. So the function of the roots are to anchor the plant to the ground, to the substrate, whatever the ground might be, right? And also do what? Besides the anchoring. And here we're looking at the anchors, right? Besides the anchoring, they usually also helping to get the water, there we go, absorb water and the minerals. And here's the picture of the hairs, root hairs. So this is the zone of division, zone of elongation, and zone of maturation. As you can see, it drastically increasing the surface area to absorb as much water and minerals as possible, because water is later on will be built into a food. More food it has, faster it will grow, outrun or outgrow the competition, so to speak. So root hairs drastically increase the surface area. Think about it. Here's the surface area of my fingers, right? That's it. That's it. That's the surface area. Now, if I'll split it, root hairs, all of that is the surface area now. So drastically increased. Same idea we'll see later on in the animals when we'll start talking about the pili and micropili in the, not pili, villi, villi in the digestive tract. Drastically increase the surface area for the absorption of any kind of food we have in our digestive system. Now, roots, some of the roots we are actually eating, the radishes, the beets, and then the carrots. And now take a look on this. This is sweet potatoes. Sweet potatoes are modified roots. Modified roots for what? For storage of the nutrients. And then, of course, humans coming in, pulling those ones out, and we eating ourselves. So yes, we are biological bullies. The plant was storing the food for itself, so it can provide the create as many seeds later on as possible, and we are digging them out and eating them. Okay, sweet potatoes, modified roots. Do you see potatoes here? No, you don't, because potatoes are not modified roots. They're actually modified stems. So sweet potatoes are actually modified roots to store the nutrients, and the regular potatoes are modified stems to store the nutrients. So now they start looking alike. We're calling it sweet potatoes and regular potatoes, but they have a different origin. That's, again, parallel, not parallel, the convergent evolution. Different parts start looking the same because they're doing the same function. So stems, already discussed the stem quite nicely, but the stem basically it's a part of the, generally speaking, the stem is the above the ground, even though there is an underground stems too. We already talked about those ones a little bit. So stem will have typically vertical one, will have several major parts. The place where the leaves are attaching, known as the node. So here's the one node, this is another node. The place between the nodes, known as the internode. Inter, again, the prefix known is literally in between the nodes. And then, of course, we have a xylem and phloem going on, and then periodically we'll have the flowers, and later on they will turn into a fruit. It will have a terminal bud, and it will have a lateral or auxiliary bud. And again, removing the terminal bud will give the auxiliary bud the green light to grow. Also remember that the plants are growing from the top up. So whenever the terminal bud is too far from the auxiliary bud, it will stop getting enough hormones to the terminal to the auxiliary bud. So that particular auxiliary bud will start developing into additional branch. So shoot systems, root systems, and then we're talking about the leaves. So already talk about the stems. You can clearly see we have 1, 2, 3, 4-year-old stem right here. Connection between the stems and the leaves. Some of them can be modified to store nutrients. See, potatoes right there. And we're eating all that particular stuff because they stored food and we're like, we'll take it from you. And then we have some more modified stems. Stem right here for the onions. Stem right here, this is the roots, and this is modified leaves. What they're modifying leaves for? For storage, and again, for sugar mostly. And this is why we have sweet onions and we are eating them. We already talked about the rhizomes. That's a modified stem, underground stem, and we have roots, and then there is the leaves coming out, and this is the ginger, for example, right? Yes. And then we already talked about the tubers, modified stems for storage. Leaves' primary function is photosynthesis. Again, remember, 6 carbon dioxide plus 6 waters plus light will give you C6H12O6 glucose plus 6 oxygens, O2. So it's essentially 666 reaction, right? 6 carbon dioxide plus 6 waters give you 6 oxygens. Yes. Ooh. So 666. Now it's a 6, you have 1 molecule of sugar produced. So it's 6616 if you go with the chemical coefficient. So mostly leaves producing going through the photosynthesis. And in this particular case, whenever you're looking at the leaf, you have the leaf blade known as the lamina. You have the veins. This is basically, essentially, it's vascular bundles. And then the veins, the vascular bundle will be attaching to the stem through the part of the leaf known as the petiole. All right. So that's pretty much it. And again, some of the leaves will be modified for the storage, and we're eating those parts. Yes, and well, actually, for that one, we modified those leaves artificially by ourselves. All of the lettuce / cabbage types, that's artificial modifications. But yes, we're eating them, even though nutrition-wise they're not very high in nutrients, but they're good for the dietary fiber, right? And then there is the tendrils, that's a modified leaves. Cacti will have the modified spines or thorns. In some cases, succulents will be storing the water. And in some cases, the bright color will attract the pollinators. So cross-section of the leaf. I know it's almost time. I'll talk about the leaves, we'll stop, and then we'll continue next time. Let me finish with the leaves first. So cross-section of the leaf. So we have a leaf cross-secting, and this is what typical one looks like. Upper epidermis, lower epidermis. Upper epidermis and lower epidermis on top will be covered by the cuticle. Quite often it would be a waxy cuticle to prevent water escaping. After the upper epidermis, we're looking at the, and then of course in the middle we have a vascular bundle. So we're looking at the ground tissue, also known as the parenchymas. So in this particular case, that parenchyma, the cells are located right next to each other, very, very close. And that layer of the parenchyma known as a palisade parenchyma, like a fence parenchyma, because they're like a pieces of fence right next to each other. The primary function of that palisade parenchyma is the actual process of photosynthesis, because it's the upper part, the sunlight hits those particular cells first, and they getting the most sunlight, so they are located right next to each other, not to lose any photon of light. So the palisade parenchyma cells are located right next to each other, primary function photosynthesis. Underneath of the palisade parenchyma, there is a spongy layer or spongy parenchyma, because now it starts having some sort of gaps between the cells. Those cells are located right next to each other in a spongy, they have a gaps. Why? Because remember, during the photosynthesis, carbon dioxide needs to go into the cell, but at the end of the photosynthesis, oxygen will be produced, which needs to be removed from the cell because it's too much. And this is what those air spaces for storage of the carbon dioxide from the outside through the special cells known as the guard cells, through the special openings within the guard cells known as the stoma or stomatas, moving into those air spaces. From the air spaces, it will be used up in the photosynthesis to produce sugars. Sugars will be moving on into vascular bundles and will be delivered throughout the body. But now oxygen from the photosynthesis will go in those spaces and then will be moved out of the leaf. So this is how essentially we're looking at, and let's take a look. Here we go. So we have upper epidermis, lower epidermis, cuticle, now Palestine Parenchyma, something also known as the mesophyll. So there is quite a few terms, and some of the students getting slightly confused. So everything, meso means in between of what? In between of the upper and lower epidermis. So basically the ground tissue. So, and on the ground tissue, we'll see the Palestine Parenchyma, sometimes referred to as the Palestine mesophyll. Don't get confused. It's pretty much just a part of the ground tissue. So Palestine, and then we have a spongy. On the lower, and that's of course in the dicot. So on the lower epidermis, it will have a special cells known as the guard cells. It's two cells right next to each other. So if the water is pumped in, they are nice and plumb, everything is good, they closed up. But now, if, for example, too much of the oxygen inside of those particular leaves, the water will be start pumped out from those cells, they will shrivel and creating the opening right there, right? So that particular opening, the oxygen will be able to leave this, the leaf, and the carbon dioxide moved in. So here the micro photograph showing you the upper epidermis, lower epidermis, the spongy parenchyma. You can see why it's called spongy, and the Palestine parenchyma, because again, like the pieces of the fence, they are right next to each other. Now, next picture shows you those particular guard cells. Again, we have two nice guard cells right there. They are live cells, and again, water pumped in. They closed up, water is pumping out, they will open up, so the gas exchange can occur. But the gas exchange, again, carbon dioxide goes in, will be used during the photosynthesis, oxygen will be leaving. And here's another picture showing you the lower epidermis and the guard cells at the, and this is all the opening known as the stoma. Okay, plural stomata. So what about flowers and fruits? What about those? You can say, well, yeah, we just talked about the flower and fruits. Well, flower and fruits are nothing else but the modified leaves. Modified leaves for what? For sexual reproduction. Okay, so let me kind of stop right here for now. And let's see.